Good morning, everyone. Thank you for being here. It's a great pleasure to open this plenary session, the first one of today. I apologize that my voice is a little bit lower. I think I spoke too much yesterday. But to announce our speaker of today, you really don't need to shout. I think his reputation and his name is already well known to most of you, if not all of the food microbiologists. We spoke about Luca Coccolin from University of Turin, a member of the executive board of ICFMH, member and a high actually contact person of EIT in University of Turin, active probably in food microbiology throughout all the continents, active in so many different areas of food microbiology, including pathogens, including spoilage, fermentation, you name it. His publication speaks for itself, right? So without any further ado, I would like to call Luca Coccolin, colleague and friend, to take the floor.
So thank you very much, Andrea, for this really inspiring presentation. Thanks a lot for the words, and good morning, everybody. Actually, it's quite amazing to see how many people are attending. I know that Athens and the nightlife of Athens is offering a lot of opportunities, a lot of possibilities, and Greeks usually they go in bed late at night, so you had the possibility to join. Greeks, for sure, you went in bed like me yesterday, pretty late in the night, but that's fine. But let me first say μεγάλο ευχαριστώ το Γιώργο, το Πάνος και την Έφη για την πρόσκληση. Είμαι πολύ ευχαριστημένος. Thanks very much for the invitation, and really, I'm very, very happy to be here sharing with you some of the findings that we had in the last 20 years. Greece has become my second family, because in my second, actually not family, but my second country, because I'm originally from Italy, but I'm married with a Greek. And let me say that this fact also is because of the people at the University here of Athens that really allowed me to collaborate and really allowed me to have the possibility to join their labs, projects, and not only George, but also the members in general. So again, thank you very much for this invitation. Today, what I'm going to be talking is a little bit specific about application of microbiomes, because when we are talking about microbiomes and when we are talking about how these new technologies can help us understanding a little bit how microbial ecosystems are working, we're always looking at, again, populations, we're always looking at ecology, but the new trend is actually go into more specific information, go deep into the possibilities that microbiome allows us to do, and go really to look the strain diversity. And this is something that I'm going to be showing, and I'm going to be showcasing with two cases that we have addressed in the last, I would say, 10 to 15 years, that is at the level of the food, but also at the level of the gut microbiome. But before that, let me just say that when we really started to look at microbial ecology with these new possibilities that we had that actually came from the technological advancements, because I consider myself, like a lot of others, probably also in this room, very lucky because we have been experiencing in the last 30 years a huge, huge improvement on the way we actually study ecology, in the way we are addressing diversity. And if you see here in this graph, the way the, actually you don't see the graph because I see the graph, but you don't see the graph. So if you see the graphs and you see how it has been developing from let me see if I can find the pointer here. Nope. Is it this one? No. Oh, well. The pointer is not actually working, but anyway. That doesn't matter. So if you see from going from plating, counting, and isolation, that I think that everybody here knows how to do it, and this is the original way we do it. Going into culture-independent methods, like, for example, DGG is the first one that we have been experiencing, the first one that entered in our labs when we tried to look at ecology. And then we switched with the advent of the next generation sequencing to RNA targeted metagenomics, and then finally to metagenomics and function target ecology studies. This allowed us to change the way we were studying and we were considering ecosystems, because actually what has been happening is that we are not anymore considering single populations, but we are considering consortia. So we have actually to think about consortia in order to discover and in order to understand how the ecosystem is working. However, what is coming is that this kind of approach, I think that I messed it up, because it's not, let me see. So it's not responding, the computer is not responding to what I'm doing, and I don't know if it is matter of. Yes, now it's working. And the pointer here is not, the pointer is not here? Sure. No. Ah, okay. Oh, wow. So this is a system where you have to point the screen, and I'm a little bit getting confused. So anyway, so the approach of all these new methodologies allowed us to understand that actually we need to go back to culturing, so we need to go back to the way we were doing things in the past, but more in a targeted way, because what we see at the metagenomic level needs to be confirmed also at the level of the plating, counting, and isolation. And so that's why the first application, let's say, of metagenomics in terms of culturomics allow us to understand what we have to do in the lab to cultivate and to be able to get out of the sample that we are analyzing, the populations that we are interested in. But what is becoming even more important is the fact that through metagenomics and through bioinformatics, we can go into genome reconstruction. So if we do things deep enough at the sequencing level, we can reconstruct the genomes of the populations that we have there, of the species that we have there, in a way that somehow we can go into a culture-independent genotyping. And this is extremely, extremely important because, as you know, and as I'm going to be repeating it at the end of my presentation, culturing introduces a lot of biases, and the ecology that we can get out of a culture-based method can be very different from the one that we are getting with a culture-independent method. And specifically for genotyping, this is, in my opinion, a very important opportunity that we have, especially when we get into specific domains, like, for instance, food safety. So this is a little bit the way I wanted to set up my presentation, and in order to show how these approaches are important, I'm going to show you, as I said before, two cases. The first one is about fermented sausages. Fermented sausages have been a subject of investigation of my lab for more than 20 years now. So the first thing that we have done with fermented sausages was to actually see how microbiome applications or microbiome studies could be applied in order to better understand what was going on during the fermentation of the sausages. And let's say, expectedly comparing the ecology reconstructed from metagenomic studies on spontaneously fermented sausages and inoculated fermented sausages, you could really see here, for instance, that when we have an inoculation, the strains that are added or the species that are added as starter culture are dominating, while if you have a spontaneous fermentation, you have much more species, you have much more diversity. This is something that we would expect. I mean, no big issue about it, no big news about it. But again, it was nice to see how, there is really this difference. What, in my opinion, is a little bit more interesting is going a little bit more down and really digging down into the metabolic activities of those species or those microorganisms through a network analysis. So here what you see is a network that we have constructed comparing or correlating the activities related to the protein, carbohydrate, and lipid metabolism with the different time points. And what it is rather interesting here is to see how the sample of the inoculated fermentation at 3 days is actually very strongly correlated with a lot of different metabolism, not only carbohydrate, mainly carbohydrate, but not only carbohydrate, while the samples that, while the samples coming from spontaneous fermentation, you see that a little bit are moved and they are not strongly correlated, or at least the correlation comes later. So the samples at the beginning of the fermentation are not strongly correlated with some of the metabolism, like in the case of the inoculated fermentation. So from this graph here, you can really say that, okay, if we inoculate, we have metabolisms that are bursted, and that are burst actually, and the fermentation that comes as spontaneous is a little bit slower. This is something important to understand, especially if we take into consider another important aspect that is actually the sensory properties of these products. So looking into volatilome, what we have seen was like a pretty strong production, especially at the end of the fermentation, of some compounds like acetic acid and ethyl acetate that are giving very, let's say, pungent notes to the sausages, but also other products like metabolites like acetoin. And if we compare those results of the volatile organic compounds with the sensory properties of those products, I mean, you can see that the inoculated fermentations, actually, they are scoring lower than the spontaneous fermentation. So the spontaneous fermentation, for almost all the indicators that we have taken into consideration, are scoring higher than the ones of the inoculated. And also, if you go into general flavor or odor, you can see how the spontaneous fermentations are actually scoring better than the inoculated fermentation. So inoculation of starter cultures is important, especially if we get in terms of safety, for instance. But we do not have to forget that when we are using starter cultures, we really need to manage them correctly, because most probably what has been happening in this study is that the company, because again, I didn't tell you that this is something that has been performed at the company level, at the industrial scale, did not manage well the starter culture. So the starter culture were too fast, they were acidifying too fast, and they were also impacting in this way the fermented sausages. This was the preliminary study that we have done within a European project. Within a European project in the Aranet area, we dig down and investigated a little bit better how specific metagenomics are actually driving the spontaneous fermentation of the Italian sausages. So here we are talking about the same fermented sausages. We are talking about the same plant. What we have done was to ferment, or actually to follow natural or spontaneous better fermentation in three different seasons, actually 3 different months, 3 different batches. And you can see how the ecology is actually different. You cannot read, but just take a look at how those colors are developing. We have specific populations that are driving, or that they are somehow guiding the fermentation in the different months. And specifically, the fermentation in March is actually influenced very much by the presence of this Lactobacillus curvatus that we did not find both in February but also in May. So also here, probably it is expected that if we do things naturally or spontaneously, probably it's going to be hard to replicate, and it's going to be hard to have exactly the same behavior. This is why we're using starter cultures, right? But it was like a kind of surprise in that, we had this great, great effect coming most probably from the raw materials that the company was using, and that was driving again, or was guiding the development of specific populations that at the end of the day, as I'm going to show you in a little bit, really impacted on the final sensory characteristics of the products. Taking advantage of the metagenomics, what we have done, as I told you before, was to reconstruct genomes. And here you see on your left the genomes, or actually some of the key genes coming from different populations of Lactobacillus, but also from Pediococcus, because Pediococcus acetylactis was actually, the Pediococcus, sorry, was actually pretty much present in the populations, or in a case, in the ecology of those products. But what I would like to highlight here is actually the difference of the genomes and the genomic setup of the Lactobacillus sakei that were present in the productions of February, March, and May. You can see that there are different profiles. You can see that that strains that are driving fermentation in the different months actually have different setup. And this is important to understand, and this is important to take into account in order really to have a kind of nice view or nice picture of what is happening inside of your fermentation and your fermented sausages, because this is something that can be used further when we go into pathway definitions. And so we reconstructed pathways. I mean, we saw, for instance, that there were differences in the production in the different months of several compounds that are really impacting on the sensory profile of the sausages. Especially here you have this ethylpropanoate, but you have also methylpropanoate and butanoic acid that actually give like a kind of that affect negatively the sensory properties of the sausages. And at the end of the day, what we can do is really, using fancy bioinformatics, go into this kind of correlations where we have the possibility to connect populations. We need to have the possibility to connect also metabolites in order to really understand what is responsible for what, or who is responsible for what. So from this nice representation, we could see that there are some specific compounds that are highly correlated with some populations. And here, what I want to underline is the rather negative impact that Lactobacillus curvatus had on the production of March. That was the one that actually the company rated lowest in terms of sensory quality. You would say, so what? I mean, we have all this information, so how can we then explore further? What we have done, basing our further investigation on these studies, was to go into the selection of starters, selection of strains for starter culture production. What we have done was to select 7 different, actually we constructed 7, you would say, synthetic communities or mixtures of mainly Pediococcus pentosaceus and Lactobacillus sakei. For the Staphylococcus xylosus, we always used one strain that was the one that, in our opinion, was the best. And what you see here is a meta-taxonomic graph where you can see that at the end of the day, when we are inoculating the starter cultures, so the Pediococcus pentosaceus, the Lactobacillus sakei, and Staphylococcus xylosus actually are the ones that we see them at the end of the fermentation. We didn't follow them throughout the fermentation. This is, again, industrial production, so we didn't do anything in the lab, but we did everything at the company level. And when we went into sensory properties of those different cultures, there were some, or consortia, there were some actually that really catch the attention of the company. And now we are in the process of really going ahead with the development of the industrial production of the starter culture in order to implement those mixtures, or in any case, those starter cultures in their production. As I said before, the second part of my talk is going to be devoted to this big topic that we have started to look at about 10 to 15 years ago. This is a very well-established statement because diet influences gut microbiome, so this is, again, established. But through the studies that we have done in collaboration with some universities in national research projects, we really could understand how diet not only, let's say, shapes the microbiome, but also shapes the diversity at the strain level. The first thing that we have done when we started this project was to go probably in the most simple, let's say, case scenario that was looking at diversity in populations that were vegan, vegetarian, and omnivores. This paper here was published by using a very low and, let's say, poor resolution method that was the DGG. However, already from this graph, you can see that there is no clusterization. There is no, let's say, differentiation of the different subjects based on the DGG profiles. So vegans are mixing with vegetarians and mixing with omnivores. Yes, probably you can see some level of differentiation here, some level of differentiation here, but again, those are very strong speculations. And what it is most important is that this picture that we got with the DGG was actually also confirmed when we applied more high-resolution methods that was metataxonomic. So this is a paper that the group of Daniele, Corina, and Francesca that is here in the audience actually developed using rRNA metabarcoding. And you can see that actually there is no difference when we look at the profiles got from the sequencing in the case of the omnivores, in the case of the vegetarians, in the case of the vegans. And this is not actually possible to correlate it if we go into the adherence of the Mediterranean diet. Here we also looked a little bit on how the diet is adhering or not to the Mediterranean diet. And as you can see here on this cake here, it was interesting to see that also some omnivores actually are highly adherent to the Mediterranean diet. So you don't need to be vegetarian, or you don't need to be vegan to have a high adherence to the Mediterranean diet. But independently of that, your profiles of the population, at least as they were described through metataxonomics, are not different. And this was a little bit surprising to us, because knowing that diet can really affect the ecology of our gut or intestinal microbiome and macrobiota, we were not expecting a result like that. Surprisingly, when we went a little bit more ahead, and so we switched from metataxonomics to metagenomics, at that point we could start to see differences. So applying metagenomics, you can see that we start to have clusterization. So the omnivores are separating more with respect to the vegetarians and to the vegans. Interesting to point out is that the genes that were driving the differentiation were the ones involved in the metabolism of carbohydrates and proteins for the omnivores. And for the vegetarians or for the vegans, we were surprised to see that genes that were involved with chemiotaxis, flagellins, were highly expressed. And this is actually something that you see here very well. Those are all genes responsible for the encoding of flagella, for instance. And this was also something that could be even more, let's say, emphasized going to metaproteomics. Metaproteomics really differentiated the vegans from the vegetarians and from the omnivores, and looking and digging down in the metaproteomic maps, we could see that, in general, pathways that were going into short-chain fatty acid production, but also group B vitamins, were all pushed pretty much, and were all overexpressed. I would not say overexpressed, but they were more expressed when we were looking at the population of the vegans and the vegetarians. We did not stop there. We were happy with the results, but going specifically, and here it comes the strain diversity, going specifically into one population that we saw very predominant, and it is also the literature that says that is very predominant, in the gut microbiota, that is Prevotella copri, we could then reconstruct the genomes from our samples, but also getting genomes that were reconstructed from other metagenomes and metagenomic studies. We could see that there is differentiation. And so strains that were present in the microbiota of vegans, vegetarians, and omnivores are actually differentiating. And this is something that is striking, especially if you see this phylogenetic tree, where you have a clear separation of the strains, actually the genomes that are coming from vegans and vegetarians, and the ones that are coming from omnivores. So again, we do not have difference at the population level or at species level, but if you dig down, then you see that there is a difference. And this somehow is confirming the fact that if you stop at the population level or at ecology level, you actually don't see a difference between vegans, omnivores, and vegetarians. But if you dig down, then you have some species that are really impacting, and they are really driving the ecology or the overall ecology of your ecosystem. I'm just going to finish here by reporting very fast two very exciting news. Not sure if Stefan has arrived. I mean, he said that he was going to be arriving late yesterday evening, but we just got the news that one proposal that we have put in this call of Horizon Europe has been funded. This is a call where we will really look into the link between fermented foods and health by looking at two main domains that are the health. So this part relates to health, this part relates to ecology. You can see that there are a lot of things that we are going to do. So this is a project that will run for 5 years, and so you will hear more about it in the future. The second exciting news is that we are moving in another dimension when we look at the interaction of gut microbiome with our health and with our body. It happened that we started to collaborate pretty well with a group of neuroscientists and neurobiochemists, and we have practically started to design an experimental setup where we can really understand how microbes are interacting with neurons. We know that the classical brain-gut axis is, let's say, bound to chemical compounds or to chemical messaging. However, we somehow hypothesize that the communication does not only happen at the chemical level, it happens also at the electrical level. So what we are going to do is, we are in the process of developing this interface where we are going to be putting together neurons with bacteria, and we are going to be looking at electrical interactions. We are going to be measuring electric signals in a way that we can really understand how the microbiome is actually influencing the neurons. And you know very well that nowadays, especially in the, I mean, can we call it post-COVID? I mean, I'm not yet at the level that we can say that we are out of the COVID. But anyway, in a post-COVID area, when we have all these issues related to mental problems, probably we can modulate the microbiome through the consumption of foods, and then have also benefits at the mental level. So those are all things that I wanted to share with you because I think that they are, again, at least for us and for my group, are really exciting news. I'm just finishing saying that the application of culture-independent methods allowed for a better description and understanding of microbial ecosystems. Again, this is, let's say, very well established. However, what I wanted to point out, especially in the second slide, is that probably we need to go back to the concept of the single strain, or in the case of the strains. And so we have to move, now that we have moved or actually we have well understood, or let's say we are trying to well understand how the community is working, probably we need to go back and we need to dig down at the strain level in a way that we can understand how the single strains are driving and are impacting or are influencing the overall, the whole microbial community. And the other thing is that metagenomics really, especially when they are done in a deep way, they allow for, again, I want to stress this term of the culture-independent genotyping. Because here we will really see the strain diversity without the bias of the cultivation step. I'm closing. I just want to thank the group. So this is the core group of my lab is Calliope, Ilario. Irene was a postdoc that did most of the work with the fermented sausages. Christian is involved in the neuron microbiome interaction. Calliope is partner not only in life, but also at the lab level. And Ilario is the one that is responsible for the bioinformatics. A long list of collaborations, specific thanks to the University of Naples. Again, Francesca is here, Daniele Ercolini, well-known scientist at the food microbiology level, University of Bolzano, University of Bari, the Bretagne Occidental University, the coordinator of the Promed Food, the RMNet project, and all the institutions that gave us funding. I'm just closing with another important announcement. For those of you that are interested, in Aberdeen next year, there is going to be the European edition of the International Association for Food Protection meeting. I'm chair. This year I have the honor of being chair of the organizing committee, so please take a look at the deadlines if you are interested to submit proposals for symposium or for roundtables. There is the 4th of October, while if you're interested in submitting abstracts for technical presentation or poster presentations, the deadline is the 17th of January. And with that, I'm thanking you very much, and I'm here for questions. Thank you.
Thank you, Luca, for the marvelous presentation and such an insightful talk that you just gave us. I think the approach that you were showing and discussing now really allows us to look at the microbiome better of a food product, but even within the human body, at least on 3 different aspects. Who is there, what can they do, and what actually do they do? I think with the several different tools that you were showing, you can actually answer those questions. Linking it to the volatile and some of the proteomes, you actually really deciphered those possibilities. Now, that brings me to the question from the audience, where Chinazo Martina Eze asked, well, she would like to understand, does it mean, in your opinion, Luca, that spontaneous fermentation is better than inoculated fermentation? That's point 1, especially bearing in mind that the spontaneous fermentation are not controlled or less controlled. And then she would really like if you could throw some light from your expertise on that. And how can this spontaneous fermentation maybe be controlled?
No, but this was, I mean, when we saw these ecological maps, so we have 3 productions in different months that actually are rather different. We were a little bit surprised because it's okay, we are in the same setups. Of course, the meat is different because the meat is different, but we have so much influence of the raw materials on the different developments that actually is somehow, I would not say destroy the principle of the spontaneous fermentation is better than the inoculated fermentation, because this is not something that we need to do. Actually, spontaneous fermentation is different because it's spontaneous. So we know very well that spontaneous fermentations are very hard to manage, are very hard to control. And so that's why we started to use starter cultures, right? But what I think it is important here to underline is that we really need to have or to be mindful when we are using starter cultures, because It's not the fact that we are using starter cultures, then our product is going to be marvelous by the quality point of view, by the safety point of view, because if we are not able to manage well the starter culture, then that is a problem. So the fact of going into the use of starter culture is like, let's say, the first step that should make the producer aware that they need to do further implementations or further checks in order to allow the starter culture to work properly. Let's say that, how can we control? I mean, the way that we have been using in the last 10 years is actually go into isolation of autochthonous or indigenous populations and try to reconstruct the populations or reconstruct the consortia or the ecology. So here we get into the very big and developing area of synthetic communities. Synthetic communities is one of the possibilities that we have. The other blowing and I would say exploding possibilities is actually the use of microbiomes. No one has investigated the possibility to store, to propagate, and to reuse microbiomes. So if we have, for instance, a product that has quality characteristics, safety characteristics that are defined by a specific microbiome, why not try to store that microbiome, propagate it, and then reuse it? This is, probably you can go back to the very classical and very old principle of the backsloping. This is exactly what we do. Because if we save part of the product and we reinoculate, actually what we do is actually reinoculate the microbiome, right? But we try, or at least we need to do it in a more, let's say, controlled or scientific way. We have a project that is going to be starting in the fall about that, and we are going to be investigating different food products, but also at the level of the environmental, for instance, and at the level of the human, how this can be done. So, again, like complicated, but I think that there is a lot of room for improvement, also by the point of view of the knowledge.
Yeah. Thank you, Luca. Any more questions from the audience at this moment? Yes, please. There in the middle.
Thank you, Luca, and congratulations on your project. Fascinating talk, really. And I have a question regarding the second part where you link with the gut microbiome. And I was wondering if there is also a direction in your field from scientists like yourselves or Danilo to close the link by studying also the relationship with the genotype, because there has been some relations, or at least very preliminary relations, saying that the human genome also is driving and interacting with the gut microbiome.
Yeah.
So I'd like to hear more.
This is another. I mean, you know what? When we started to look at the, let's say, influences of diet, we needed to make assumptions and we needed to simplify the system for sure. I mean, we know, for instance, that the diversity here, I don't know how many we are, but probably all of us have a different microbiota and a different, probably, no, I don't know how different is going to be the microbiota, but the microbiome most probably is going to be different. So for sure, there is an element of complexity that we didn't yet, or we were not able yet to control. When we get into genotyping, I mean, I agree with you that this is an important aspect that has to be taken into consideration. There are some very famous studies about, twins and things like that. But again, when you are moving to population studies, this is something that is very, very complicated to control. What I can tell you is that experiences have shown that when you perturb a microbiome is changing. If you take away this perturbation, microbiome goes back. The typical example is about probiotic supplementation. So if you do, the probiotic supplementation, and you go now, these days is very hot talking about superfoods. I mean, everything is a superfood. So, but when you go on intervention with a superfood, you see a difference. Microbiome is changing. When you take away, microbiome goes back. How is it going back? Is there a memory? Where is this memory kept? Is it at genomic level? So, this is something very, very interesting that, but again, at population level at the moment is very, very complicated.
There are a number of other questions, but I think we are running a little bit out of time. So I'm looking at the organizers. Do you allow a few more questions, or do you think that we have to continue? Well, maybe one short question there, because I see next to George, and I think George takes responsibility for it.
Konstantin, Jenny Georges, University of California, Davis. I'm kind of a prehistoric creature telling you prehistoric stories. And I'll go back to 1974, thanks to a lady who had diarrhea, staphylococcal food poisoning from Italian dry salami. We embarked in 10 years of studying the ecology of those products. And one of the first things we identified was the international cultures could not work simply because they could not adopt to that environment. So eventually then we studied the ecology, isolated the bacteria, developed the technology to prepare the cultures, and eventually were able to make Italian dry salami in the desert by controlling the environment. So environment is very important then because it drives which one is going to come first, which one is going to be second, which one is going to give the flavor compounds. That's all. Thank you.
Thank you. Thank you.
Okay, thank you all. I think the discussion could continue, Luca. That just shows how
I'm
inspiring the
here.
talk was, but don't miss the opportunity to talk to Luca just before. And before Luca leaves the stage, I have something for you in the name of the organizing committee.
Wow, thank you. So my acknowledgements at the beginning were well, gave a good result at the end.
That said, I will leave the chair to the next people.